Okay, back to Wildfield. So we've already discussed this a little bit, but I wanted to add in an extra layer. So as we mentioned when we were comparing Wildfield and Confocal, Wildfield, you're illuminating the full field of view, and you're not getting that nice clean optical section that you are on a confocal microscope. But we can create confocal-like images from Wildfield data, and we do that using a method called deconvolution. So what this is a mathematical restoration method which can increase our resolution in all three directions and increase our signal to noise as well. So as the image goes through the microscope comes what we call convolved, so we lose a bit of the detail. And what deconvolution is doing is restoring that back mathematically, back to what it would have looked like originally if it hadn't gone through the microscope in the first place. The requirements for this is that we need ideal sampling on the sample. So again, we'll go through the Nyquist-Shannon sampling on Thursday. And ideally, we want a Z-stack of the data as well. So we need a 3D volume. So there are a number of different software packages available. Some of the vendors have them on instruments. But we have 2 Huygens professional licenses available within the platform for people to access, and that will work on Wildfield, Confocal, Airiscan, and also Lightsheet data. It's GPU accelerated, so it goes much faster, and we can also run processing in batch as well. So what I should say is we're using a model of how the light behaves going through the instrument, and the mathematical restoration uses that model to kind of reassign the light that may have been coming from another focal plane back to where that light originated. And again, this is an example here of deconvolved versus Wildfield data. Okay, let's move down the scale. So we've gone from kind of the tissue organ level down into cells, organelles, and proteins, and there's a lot to go through in this section. So we're going to cover TIRF microscopy, confocal microscopy, a group of methods called the F experiments, and then we'll go through some of the super resolution methods as well down here in green. Okay, so TIRF microscopy. Normally in a wide field illumination, we are illuminating our full field of view, as mentioned multiple times, and that light is transmitting up through our sample. This therefore can create very fuzzy images as we're exciting all of the molecules within this volume that we can see. What we're doing in total internal reflection mode is we're hitting a specific wavelength that we're transmitting the light through. It's actually going to bounce off the cover slip. So here we're getting total internal reflection off the cover slip back into the objective. And that means we produce something called an evanescence wave, which essentially illuminates only the first 100 nanometers past the cover slip. That means all these molecules up here aren't getting excited by the fluorescence. And that means we get a really nice, sharp image with nice signal to noise, because we only have essentially a confocal section just past the cover slip. So it's not going to work if you're looking for things necessarily, say, within the nucleus, but if you're looking at cytoskeletal components or things that are in the cytoplasm, then TIRF microscopy might work nicely for you. So there's an example there. Some applications are things like live cell imaging, calcium flux. I mentioned the cytoskeleton. It's very often used in some of the super resolution microscopy approaches as well. Okay, confocal. So there's a lot that you can do on a confocal microscope. Again, it's probably a 1-hour lecture in itself. So we can make use of automated stage functions for doing things like 3D acquisitions or multi-point visiting experiments or tile scanning if you have large samples. We can undergo dynamic imaging, so live cell imaging. Again, the F experiments that I mentioned, looking at protein dynamics or protein-protein interactions. things like photoconversion. And then there are a number of specialized detectors that are available on confocals as well. So we'll go through in the super res section one of the detectors for doing super resolution microscopy, and also we can do spectral detection as well. So this is an example of the automated stage function. So here, what we can do is take a single field of view on, say, a reasonably high magnification lens, but if our sample is much, much larger, we can actually use the confocal and the stage functions to scan across that sample, image it at fairly high resolution, but again, take in the entire sample. And again, this is a retinal home mount from one of the groups here at the university. I mentioned spectral unmixing. So this is a specialist detector. So there's a couple of ways we can do this. On some of our systems, the Zeiss systems within the platform, we have this multi-channel array. So this is made up of 32 individual detectors. We can take the light coming back from the sample, and we can use a grating to actually separate that out across the visible light spectrum. And we can read that out across each one of these individual detectors. And what that allows us to do is to build up a spectrum of what the molecules would look like. Normally, you're using individual color controls to come up with the individual spectrum. Once you have those in place, then you can start to put together a large number of fluorophores into your sample. So in this case, we had one scientist doing 9 channels and using this approach to essentially spectrally unmix each of these 9 different channels. And then he was going in and identifying individual cells based on their different stains for each of these markers. So very, very useful. This detector allows you to do it in one shot. There are other detectors which again have a diffraction grating or a prism-based approach for opening up the spectrum. And then there's a variable slit here, which they can vary and kind of scan across the wavelengths. Obviously, this takes a lot longer than the one-shot spectral array detector. We mentioned live cell imaging. So within the platform, we have one spinning disk confocal. So unlike a standard confocal where we have the laser beam scanning across our sample, building up the image as we go, in this case, as the name suggests, we have a disk with lots and lots of pinholes within it. And that has the benefit that we can image really, really quickly. So this is a camera-based readout. This disk is spinning really, really quickly. It means that not all your sample is illuminated at all times. The downside is it's not really that applicable to thicker samples because you get too much out of focus light that bleeds through between these pinholes. Spinning disk systems are very, very useful for imaging. If you're imaging things like mitosis, they are very, very sensitive to light. Samples which are very, very phototoxic. So in this case, we've got an example of some cells undergoing mitosis. The spinning disk systems are being a little bit usurped at the moment by a system called a lattice light sheet. Unfortunately, we don't have one of those in our platform at the moment, but yeah, this is another possibility in terms of systems if your sample is very, very sensitive to light.